we're going to take a look at some different types of alkanol reactions, starting with dehydration, which we can call elimination. Now, with dehydration, the products formed depend on the conditions used. For example, alkenes are formed in the presence of sulfuric acid. Now, phosphoric acid is better since it does not produce as many byproducts. The correct temperature is also important because hot is used for primary, warm for secondary, and cool for tertiary. And then alcohols actually lose a water molecule in the process of dehydration. Now, with oxidation, the products depend on the type of alcohol used: primary, secondary, or tertiary. For primary and secondary, a double bond between carbon and oxygen replaces a single bond between carbon and hydroxide. But this bond will either be at the end of the carbon chain, which would make it an aldehyde, or in the middle of the carbon chain, making it a ketone. Aldehydes can be further oxidized to form carboxylic acids, and tertiary alcohols will not oxidize. Now we come to substitution, which is usually not very important for the alcohols except for halogenation using phosphorus pentachloride. So substitution may be used as an identifying test for the alcohol group. Then a reaction with active metals is just what it sounds like. When alcohols react with active metals, and in that they release hydrogen. And so this also may be used as an identifying test for alcohols. Now, esterification is a reaction between an alcohol and a carboxylic acid. And this forms an ester link, which looks like this. And this holds two carbon chains together. Now, the conditions required for this reaction are concentrated sulfuric acid and elevated temperatures.
Biological classification systems. Organisms are divided into three domains. Eukaryotes belong in eukarya. Prokaryotes are divided into the domains archaea and bacteria. Beneath domains, there are other rankings that organisms are divided into so that we can keep all the organisms organized and see how they're grouped together, see which organisms are related to other organisms, and from that determine which characteristics they may have in common. So let's look at the rest of them. And after domain, you've got kingdom, phylum, or division. And phylum is going to be referred to in eukarya, where divisions are going to be referred to more in the archaea and bacteria domains. And then you have class, order, family, genus, and species. So species is the most specific, where the domains are going to be the most general. The domains are going to hold everything that fits under that category, where each species is going to hold just a small, very specific organism. And organisms are named scientifically with binomial nomenclature in eukaryotes. So binomial nomenclature is used to name eukaryotic organisms. And that means that you use these last two, bi meaning two, these last two names to name an organism. So you would use the genus and species of an organism to give it its scientific name. You will have a more common name most of the time for a creature or an organism, but there is also a scientific name where you will be able to see what the species and genus is, and from that see which family, order, class, phylum, kingdom, and domain that organism would fit into. A species is defined as a population of organisms capable of interbreeding with each other and producing viable and fertile offspring, which means that they are producing offspring that is both alive and healthy, and it's able to reproduce in itself. If the organisms are producing offspring that aren't capable of reproducing, then that species is not going to be able to continue, or that organism's growth isn't going to be able to continue, and it's not going to be considered a species in itself. So each species is only going to consist of organisms that can breed with each other and produce viable and fertile offspring that can also reproduce. So let's look at an example. And before we do that, I'm going to give y'all an acronym to help you remember the order of these. So you've got your 3 domains, and then under each domain you're going to have kingdom, phylum, class, order, family, genus, species. Well, it may be hard to keep track of what order those go in. So here is a helpful little saying that should help you. Keep ponds clean, or frogs get sick. And if you look at the first letter of each of those, they will remind you what the order of our rankings are. Kingdom, phylum, class, order, family, genus, species. So let's go through and see if you can figure out what this is a classification of. This organism is in the kingdom Animalia, which is going to be all animals, and it's going to be in the phylum Chordata, which is going to be organisms with spinal cords, and we're going along with this and this. Look wherever you like, but we want to remember which order we're going in. So I have them listed here, but if you want to say the acronym in your head to get used to the order of the rankings, then you can definitely do that. So kingdom, phylum. Next we've got our class, Mammalia, which is going to be all mammals, all organisms that are able to produce milk for their young and give birth to live young in most situations. We have a couple of egg-laying mammals. And then you've got your order, Carnivora, which is going to be carnivores, animals that can eat meat. Family is Felidae. Genus is Felis. And species is Caddis. So your scientific name is going to be found using binomial nomenclature. You know this is a eukaryotic organism because it's in the kingdom Animalia, which is a eukaryote, so it would go into domain eukarya. So binomial nomenclature means we use these last two. And using our genus and species, our creature is going to be known as Felis caddis. And what do we think that is? If you guessed a house cat, you are correct. This was the classification of a house cat. And Felis kind of sounds like feline, which you've probably heard before. And caddis sounds like cat. So a house cat belongs to the kingdom Animalia, phylum Chordata, class Mammalia, order Carnivora, family Felidae, genus Felis, and species Caddis, giving it the name Felis caddis and the common name of house cat. This would be the common name. So, whenever you are looking at an organism and you see some Latin words like this, 2 Latin words together, you are probably looking at the scientific name for that organism. And if you know the genus and species of an organism, you can figure out which other organisms are members of that group and figure out what they might have in common and slowly build up and figure out what family, order, class, phylum, kingdom, and domain that organism is in. Or the more things that you know about an organism, the more you can break it down and put it into different kingdoms, phylums, classes, orders, a family, a genus, and a species. So this classification system is used internationally so that Anywhere in the world, if you saw this Latin name, any scientist, whether they spoke English or another language, would be able to know what species that scientific name was referring to. And this classification system is used by all scientists. That way, all scientists will be on the same page if they're trying to classify a new organism or figure out which rankings an organism that they find might fit into, to see if it is indeed a new organism or something that's already been discovered and they're just trying to classify it. Whenever you are trying to remember the order, keep in mind this handy acronym, keep ponds clean or frogs get sick, and that should help you keep these guys in order. And remember our 3 domains, eukarya, archaea, and bacteria. And the most important thing to remember about your biological classification system is that it's there to classify these organisms. It's there to keep you organized. DNA, or deoxyribonucleic acid, contains all of the hereditary or genetic material in humans and most other organisms. So the DNA is what lets people know what genetic material is in a person or is in a certain creature or other organism. Now each cell in an organism has the same DNA. So if someone were to get a piece of your hair and a fingernail and a piece of skin and look at it very closely and look at the DNA in all 3 would be the same, and it would let whoever was looking at this know that all 3 of those articles came from the same person. This is the same for other organisms. Each cell in the organism's body is going to have the same DNA pattern so that you'll be able to tell that it came from the same creature. Most DNA is found in the nucleus of a cell. There is some DNA in the mitochondria, and then it is known as mitochondrial DNA, or it would look like this if you saw an abbreviation for it. So if you see the little MT before DNA in the abbreviation, that's because it's mitochondrial DNA and it's found in the mitochondria instead of the nucleus. But most DNA is going to be in the nucleus. Now, that important hereditary information found in DNA is stored as a code made up of 4 chemical bases. We've got adenine, guanine, cytosine, and thymine. And each one of those bases is only going to pair with one other base. They can't all pair up with any of the other 3. So adenine pairs with thymine, and guanine pairs with cytosine to form base pairs. And a way that I remember that is that the ones with the straight lines in their abbreviation letter, the A and T that can be made with straight lines, go together, and the ones that require a curved line go together. And that helps me remember that adenine pairs with thymine, these straight lines to form A and straight lines to form T, and guanine goes with cytosine because of the curve shape that it takes to make each of those abbreviation letters. The sequence of the adenine, thymine, guanine, cytosine pairs determines how the organism builds and maintains itself. So it's important to think about this. Not every organism or every person is going to have the exact same sequence of base pairs. And you might think of the alphabet. We use 26 letters to form all of our words and sentences, but the way that they're arranged, the sequence of those letters, is what lets us know what word is being communicated, what sentence is trying to be communicated. And each word has a different meaning, even though it's made up of the same 26 letters or some subset of those letters. So it works the same way. You're going to have some sequence of adenine, thymine, guanine, cytosine, and those base pair sequence is going to let you know, or let a scientist know, what that organism does to maintain itself, what kind it is. Is it functioning properly? Is everything in the right order? What does that order tell us? The same way that seeing CAT tells us that that is the word cat, and it's talking about a mammal with pointy ears and whiskers and a tail that's kept as a pet. Let's look at the next part. Each base pair, so our adenine-thymine pair or guanine-cytosine pair, is going to attach to a sugar molecule and to a phosphate molecule. And once those attach, once a base pair attaches to the sugar molecule and phosphate molecule, it forms a nucleotide. And these nucleotides form two long strands that spiral into a double helix. That is the shape that DNA takes on. And I did my best to give you all an example of a double helix. It kind of spirals around. If you were to curl a ribbon around my pen, it would form that kind of double helix shape we're talking about. And you should think of a twisted ladder with the base pairs as rungs. So if we were looking at this picture here, this would be our double helix shape that's spiraling around, and each one of these little rungs on the ladder would be a base pair. So we would have adenine and thymine together, and they would be paired up. And then here you would have guanine and cytosine paired up. And then you would have adenine and thymine, guanine and cytosine, all the way down the ladder, where you've got your sugar molecules and phosphate molecules that bound to your base pairs, making up the sides of the ladder here. And you just have this long DNA chain, and then the sequence that you see is going to let you know how that organism builds and maintains itself, because not every creature's DNA is going to be the same. Everyone has a very unique DNA pattern. DNA can replicate or make copies of itself by splitting the ladder in half. So if we're looking at this and we just decided to go through and cut this ladder in half, we would be separating those base pairs all the way down. So then you have AG, AG, and any other base pairs you've got. So we know we've got adenine, guanine, and because we know what's on one side of the ladder, each strand or half of that ladder serves as a pattern for duplicating bases. Since we know we've got adenine here, we know the thing that has to pair with it is thymine. If we've got guanine, cytosine has to pair with it. Since in DNA, adenine and thymine always pair together and guanine and cytosine pair together, you will know, or the cell will know, okay, I've got this DNA, I've only got half of it, but it's a pattern. If I have adenine, I need to add thymine to the other side. If I've got cytosine, I need to add guanine to the other side. So the cell will know, based on that half, that strand that it gets from DNA, how to make a complete DNA molecule and be able to form that double helix and complete the DNA strand. And each new cell needs an exact copy of DNA from the old cell. Because remember, each cell in an organism has the exact same DNA. So if the DNA is replicating itself, it needs to be giving an exact copy to a new cell so that this can continue, and the organism is going to continue to have the same DNA in it. So you will see a lot about DNA in biology, and it's important to remember that DNA is the hereditary or genetic material. It's usually found in the nucleus. And remember which bases pair with which. Your straight lines in adenine, A, pair with your straight lines in thymine, T, and your curve G for guanine goes with your curve C for cytosine. DNA is the hereditary or genetic material that is usually found in the nucleus, sometimes in the mitochondria. The most important thing to remember is that it's where all the genetic material is going to be found. It's a big identifier for a cell or for an organism. Enzymes. Enzymes are highly selective catalysts that greatly accelerate the rate of reactions. So they speed up how quickly a reaction takes place. Most enzymes are proteins, but some catalytic RNA molecules have been discovered. So there are RNA molecules that will catalyze reactions and help speed them up in the same way that these proteins will, and so they both work as enzymes. In enzymatic reactions, the molecules at the beginning of the process are known as substrates. During the reaction, these substrates are turned into different molecules known as products. And at the end of the reaction, the enzyme will be unchanged. The enzyme may have to change shape a little bit to bind to the substrate and finish the reaction, but the enzyme itself will be unchanged. It will not have turned into different products. It will not have gotten bigger or smaller. It will remain unchanged. Enzymes work by lowering the activation energy of a reaction, drastically reducing the reaction time. If you had to build up all that energy to complete a reaction normally, it would take a really long time. Well, whenever the enzyme works on a reaction, it lowers the activation energy and makes the reaction happen a lot faster. In fact, a lot of our metabolic processes would not be fast enough to sustain life without enzymes there to speed up the reactions. Enzymes are highly specific for their substrates. That means that each enzyme will only bind with specific substrates that fit with its active site. So each enzyme has an active site. Here we've got our enzyme. To give you an example, this area would be the active site. So both here and here, this open area in the overall shape of the enzyme is the active site, and each enzyme has an active site, and only substrates with a similar shape that would be able to fit in there and bind without the enzyme changing shape too much will be able to fit with that enzyme and complete a reaction. So this would be your substrate. And this is where the substrate would be coming into contact with the enzyme. Here you can see the enzyme and substrate have bonded, and now they have formed the enzyme-substrate complex. And you can see that the enzyme changed shape a little bit to bind to that substrate. And then in this next phase you have the enzyme product complex, where the substrate has started to turn into two different molecules and separate from each other. And then in this last part, you've got your enzyme remaining unchanged, and the products, two separate molecules that the substrate has been turned into, will leave the enzyme. So you can see the enzyme remained unchanged, and the substrate was turned into two separate products. And going through this stage is going to lower the activation energy of a reaction. Enzymes can be affected by inhibitors, which are molecules that decrease enzyme activity, and they can be affected by activators, which are molecules that increase enzyme activity. So where you have enzymes that are raising the rate of speed of a reaction or lowering the activation energy needed, the inhibitors are going to slow that process down, and the activators are going to speed that process up. So where the enzyme is acting on a chemical reaction, these other molecules can also interfere with the chemical reaction and either speed it up or slow it down. The main thing to remember about enzymes, is that these are highly selective catalysts, and they are going to increase the rate of a reaction by lowering the activation energy needed, and at the end they are going to remain unchanged. Eukaryotic and prokaryotic cells. There are a lot of differences between eukaryotic and prokaryotic cells, and there are a few similarities. So we've set up this chart to kind of compare and contrast each of them so you can see the differences between the two. Cell type. Eukaryotic cells are usually multicellular. They're going to come in a bundle of cells, where prokaryotic cells are usually unicellular. There'll just be one single cell standing by itself. Examples would be animals and plants are composed of eukaryotic cells. There are a lot of cells that make up each animal or plant. Bacteria and archaea are prokaryotic cells. Usually it's just one cell standing by itself. Nucleus. Eukaryotic cells have a nucleus, and prokaryotic cells do not. They do have a nucleoid, which has nuclear material just kind of floating in the cytoplasm, where the eukaryotic cell has a cell membrane and has a nuclear membrane and keeps the nucleus separate from the rest of the cell. Number of chromosomes. Eukaryotic cells are going to have more than one. The prokaryotic cells are just going to have one, and it's going to be a plasmid and not a true chromosome. Lysosomes and peroxisomes. Yes, eukaryotic cells will have those, and they will be membrane bound. Prokaryotic cells will not have membrane bound lysosomes and peroxisomes. Microtubules. Yes, eukaryotic cells have them. Usually no on the prokaryotic cell, but rarely they will have microtubules, but it's very rare. It's more likely that you'll come across a prokaryotic cell that will not have microtubules. Endoplasmic reticulum. The eukaryotic cell has one, and it's got parts that are both smooth and rough endoplasmic reticulum, each part functioning a little bit differently. Prokaryotic cells do not have an endoplasmic reticulum. Genetic recombination. Eukaryotic cells are going to reproduce by meiosis and the fusion of gametes. Prokaryotic cells it's partial and unidirectional reproduction. It just kind of transfers DNA to another cell. So it isn't as if the cell is splitting and giving half of its DNA matter to something else. It's just transferring some to a new cell. DNA wrapping on proteins. In eukaryotic cells, the DNA wraps around proteins called histones. In prokaryotic cells, there are multiple proteins that fold and condense the DNA, and once it's folded and condensed, it's supercoiled and wound around tetramers of HU protein. Mitochondria? Yes, eukaryotic cells have them. No, prokaryotic cells don't. And you can see the eukaryotic cells are going to be a lot more complex than the prokaryotic cells. So they do have more things in them, and the prokaryotic cells have less inside of them. Flagella. These are microscopic on eukaryotic cells. They're membrane bound, and they are usually arranged as 9 doublets, so 9 little pairs of flagella surrounding 2 singlets. On prokaryotic cells, they are submicroscopic, so even with a normal microscope you wouldn't be able to see them, and they're composed of only one fiber, where this one has 9 sets of little fibers surrounding 2 singlets. Cytoskeleton. Yes, the eukaryotic cell has a defined cytoskeleton. With prokaryotic cells, it may or may not be present. You may have some little bits that give it a skeletal shape, but usually there's not much cytoskeleton in a prokaryotic cell. Ribosomes are going to be present in both. They're going to be larger in the eukaryotic cells and smaller in the prokaryotic cells. The Golgi apparatus will be present in the eukaryotic cell and absent in the prokaryotic cell. Chloroplasts. Yes, they will be in eukaryotic cells if it's a plant. Chloroplasts are what holds the chlorophyll. So in plants, you've got chloroplasts to hold the chlorophyll in. In prokaryotes, No, you do not have chloroplasts. The chlorophyll is just kind of scattered in the cytoplasm, little dots here and there to help absorb the sunlight. Vesicles, yes, in both the eukaryotic and prokaryotic cells, and vacuoles, yes, in both. So here's what they have directly in common, that they both have vesicles and both have vacuoles. Now, in some of these other characteristics, they do have something similar. They both have ribosomes, but one is larger and one is smaller. They both have nuclear material, so the eukaryotic cell has a defined nucleus, and the prokaryotic cell has the nucleoid, which has some genetic material in it. And then cell size is going to be 10 to 100 micrometers, which is really small, and prokaryotic cell is going to be 1 to 10 micrometers. So you can see that the cell size on eukaryotic cells are going to be about 10 times larger on average than these tiny prokaryotic cells. There's a lot of information here, and there's a lot to remember about prokaryotic and eukaryotic cells, but as you learn about each one of these things individually, it will be easier to remember which one goes with which, and you can always refer back to this chart to kind of keep track of what is in a eukaryotic cell, what characteristics the eukaryotic cell has, and what all is inside of a prokaryotic cell and what characteristics it has. Feeding among heterotrophs. Heterotrophs obtain their energy by feeding on other living or dead organisms. So heterotrophs cannot create their own nourishment. They must get it from other organisms. And there are lots of different ways that they do that, and the ways that they do that classify them as certain types of organisms. So heterotrophs may be carnivores, which are animals that eat only other animals. Examples would be lions or tigers. Heterotrophs can be herbivores, which are animals that eat only other plants. So examples would be deer, elephants, koalas. These animals only eat plants. They're not going to eat other animals. Now, on both of these, there may be rare exceptions where a carnivore may eat some kind of vegetation, or an herbivore may eat some other animal, but it's very rare, and so they're still classified either as a carnivore or as an herbivore, because what little bit they eat of the opposite kind of food is just a rare exception. It's not something they need for their diet. Now, the next one, omnivores, they eat both plants and animals, and they require both to maintain a healthy balance of nourishment in their bodies. Examples would be pigs, bears, raccoons, and people are also omnivores. We eat both plants and animals, and we require both, or sometimes people take vitamins to supplement if they don't eat both, to maintain a healthy system. The next example are detritivores. And detritivores eat decomposing organic matter. So the detritivores would be the decomposers in a food chain. And examples would be millipedes, wood lice, and slugs. Next we have parasites. And parasites are going to obtain nourishment from a host organism without killing it, but definitely harming it. The parasite is going to take something away from the host organism so that the parasite itself can be nourished. An example would be a tapeworm. A tapeworm gets inside a vertebrate host, such as a mammal. People can get these, and other mammals also get them. And the tapeworm is ingested somehow, and then it settles in and it starts taking the nutrients from the food that the mammal or the other vertebrate is eating. And everything this organism eats is going to the tapeworm instead of going to themselves, and so the organism starts to get malnourished while the tapeworm flourishes and grows and takes up more and more space inside that organism's body, while the organism is not being actually nourished even though it's eating. So they are known as parasites. Next we have scavengers. Now scavengers actually search out and consume dead animals or plants. So instead of it just being something they happen upon, it's something they search out. Scavengers want to eat dead animals and plants. And examples would be vultures and blowflies. And some of your herbivores, carnivores, omnivores may eat a dead animal or plant from time to time if it has nothing else to eat, but it's not searching it out. So it would be like if either of these, if a carnivore ate plants or an herbivore ate animals, it would be a rare exception for them to want to eat the dead animals or plants. But scavengers search those out as their primary source of food. And last we have coprivores. And coprivores eat animal feces. So dung beetles and flies would be examples of that, but there are other animals like young elephants or rabbits or hamsters that eat other animal feces to get that last remaining organic matter out of it, to try to get the last nutrients they can. And so animals that eat other animal feces are known as coprivores. So as you can see, there are lots of different types of heterotrophs, lots of different other organisms, living and dead, that heterotrophs can feed on, and they are separated by which kind of living or dead animal they eat, or animal products. But the main thing to remember about heterotrophs is that they aren't able to make their own energy. So each type of heterotroph is just telling you how they're getting their energy from another living or dead organism. Gene mutations. Gene mutations have several different types. So one would be a missense mutation, which is where one segment of DNA is substituted for another segment of DNA, which tells the gene to make a certain amino acid. And if it's a missense mutation, then it's telling it to make the wrong amino acid. It did code for an amino acid, but the wrong one, and so this could mess up the chain. You could also have a nonsense mutation, where again a segment of DNA is substituted for the wrong segment of DNA, and it doesn't code for anything. It doesn't code for an amino acid at all, and so the chain ends there. And so it cuts that gene short and could affect the gene function. You also have insertions, which is where a segment of DNA or a base pair is inserted where it doesn't belong, and it alters the arrangement of base pairs, and it could cause the chain to end early, but it's definitely going to change what the chain does, and the function of that gene, that segment of DNA, is going to be changed. You have deletions as well, where a base pair is just deleted, and then most likely the chain is not going to function anymore, and that genetic function is going to fail. So there are different types of genetic mutations. You can have substitutions, where you just have this point substitution, this one area where a segment is changed. You can have insertions, and then you can have deletions. And sometimes this leads to just that one small incident, and sometimes it leads to replication, where there are lots and lots of mutations instead of just the one mutation. Some causes for gene mutation are environmental causes, such as chemicals that you either ingest or that touch your body, radiation, and ultraviolet light, such as from the sun. If you have too much, or sometimes just any amount of some of these things, especially chemicals, any amount of certain chemicals, and then too much radiation or ultraviolet light can make your cells mutate. And it isn't something that happens always, and it isn't something that you can say this will specifically happen, but these are environmental causes linked to gene mutation. Biological causes would be errors made during mitosis and meiosis, and these would be something that certain mutations that might occur in a child whenever the two parents are creating the child. So when that child is forming and mitosis and meiosis are going on, any kind of hereditary mutations are going to be created in that offspring at that time. So you can have environmental causes and biological causes for these gene mutations. And then results of the mutations. Well, the original course of that DNA sequence is altered. So now something else is going to happen. There could be no effect. It could just end. It could not have any effect at all if that DNA chain ends and that function isn't carried out. Maybe it wasn't something very important, and so there's no effect on the organism. It could alter the product of a gene. So the gene's product could come out differently than it was supposed to. Or it could prevent the gene from functioning properly or completely. So maybe the gene's product doesn't get shipped out at all, or it ships out something that it's not supposed to ship out, or it ships out part of something. And the results can be neutral. For instance, a gene mutation in your eye color. You can have brown eyes, blue eyes, green eyes, and the mutation doesn't harm you or benefit you. Generally, it's just going to be a difference, and so it would be a neutral mutation. You can have harmful mutations, such as that causes Down syndrome. You've got an extra chromosome there, and that mutation causes physical and mental problems. And then you can have beneficial mutations, such as that causes sickle cell disease. In the area where sickle cell disease developed, people that had the sickle cell disease were able to fight off malaria better, and so it was a survival benefit to have that mutation. So gene mutations can be caused lots of different ways, and there are lots of different types of mutations, and while most of them have little benefit or harm to us, there are some that can harm or benefit us, but genetic mutation is something that's necessary because it's what keeps people and other organisms evolving and becoming more and more advanced organisms. Genes. A gene is a molecular unit of heredity of a living organism. A gene is the basic instruction, a sequence of nucleic acids. So a gene is going to be the instruction, it's going to be the sequence of nucleic acids that tells the cell to code for a certain protein or for a certain RNA chain. It isn't just one little thing, it's a sequence that gives an instruction to the cell. While an allele codes for one variant of that gene. So if you've heard someone say, oh, she has really good hair genes, or she's got the gene for blue eyes, that's not exactly true, because everyone has a gene for eye color or hair color, and for every other trait that you have. But what determines what color your hair is or what color your eyes are is going to be the alleles for that gene. So there are dominant alleles and recessive alleles. And if you have even one dominant allele, then you're going to have the dominant gene. If you have two recessive alleles, then you will have that recessive gene. For instance, if you have two parents that have brown eyes, you could still end up with blue eyes because blue is a recessive gene. So both of your parents had one dominant and one recessive brown and blue eye color allele. The brown would have been prevalent in both of them. But since they both had that recessive blue allele present, you could still end up with blue eyes because of the recessive alleles combining. And I know that all sounds a little confusing, but it's just to tell you that each gene doesn't determine something all on its own. It's the alleles that are the variants of that gene that determine what the outcome of each gene sequence is. Genes do specify all proteins and functional chains. So they specify how these are going to be created, the functional RNA chains. Genes hold the information needed to build and maintain an organism's cells and pass genetic traits to offspring. So where I was talking about two parents that could have brown eyes and still pass on the blue eye color, it's because of the alleles that they carry, whether dominant or recessive, whether you can see it as a physical trait or not. Your parents carried genetic traits and they passed those on to you. And some of your physical traits may not all be the same as one or both of your parents, but the genetic traits that you got did come from the alleles that they had combined and mixed together. So you took some of your mother's alleles and some of your father's alleles, and when those combined, you ended up with different alleles and different genetic traits of your own, but they both came from the genetic traits your parents had. Some examples are eye color, number of limbs, and blood type. So some genes you can see right away manifested, such as eye color or number of limbs. But blood type is something that you would have to do a little more scientific research to discover. You can also search for certain markers in your body for diseases such as Alzheimer's or other hereditary diseases. Those can be tested for, and your DNA will show, your genes will show if you have a likelihood of getting that disease. And so that's definitely not something you can see just by looking at someone. It's something that you would have to do more research into. But it's possible by looking at the genes in your body. Now, for a gene to create or code for its protein, it must go through transcription. And transcription is where single-stranded messenger RNA or mRNA is created that is complementary to the DNA from which it was transcribed. And we'll look at that over here in just a minute. But basically, you're starting with DNA and you're transcribing it into the messenger RNA. And then the messenger RNA is going to bring the message elsewhere in the cell. So next comes translation. That messenger RNA is used as a template for synthesizing a new protein. Transfer RNA, or tRNA, brings the amino acids and anticodon necessary for protein synthesis. And something to remember is that genes are read 3 nucleotides at a time in units called codons. So you look at the 3 nucleotides that are coding for this one protein, and they're going to be transcribed onto messenger RNA, which is going to travel elsewhere in the cell, and transfer RNA is going to bring the amino acids and the anticodon that match up with that set of 3, with that codon, and let the protein be created. So let's look at that over here. Let's pretend this gene sequence came from a strand of DNA. And remember, your bases are adenine, guanine, cytosine, and thymine. So we are going to say it's going to change from DNA to the messenger RNA. So we're transcribing here. And each base must be transcribed. So here we've got our transcription. So the guanine is going to transcribe to cytosine, cytosine to guanine, thymine to adenine, cytosine to guanine. There we go. Thymine to adenine, and thymine to adenine. So the messenger RNA is going to carry this sequence. So you can see how it isn't exactly what was on the DNA, it's what base would have paired with the DNA. So it's kind of like a code you've got to sit here and figure out. You've got to decode what the messenger RNA says. So the messenger RNA has the pairs that would have gone with the DNA, and so in comes your transfer RNA, which is going to have its amino acids and the anticodon that's necessary for each of these. So each set of 3 is a codon, and it gets transferred over. And so now the transfer RNA is going to come in, and it's going to bring what we need. So it's going to say, okay, we had cytosine, so now we need the guanine. I guess we should really point this way. And then it's going to bring cytosine back to pair with the guanine, thymine back to pair with adenine, cytosine back to pair with guanine, thymine to pair with adenine, and thymine to pair with adenine. So now, in a different part of the cell, you've got these codons, and they're gonna code for a certain protein. And this is what happens in the cell. So this part would be your translation. So you've got strands of DNA. Part of the cell says, oh, I need a certain protein. Well, this codon codes for one of the proteins you need, and this codon codes for one of the other proteins you need. So the DNA is going to send it. The messenger RNA is going to hold the transcription, which is where it's changed to what the pairs would be. What pairs with each base is going to be the transcription that goes on the messenger RNA. And then the messenger RNA travels off into the cell to where the protein is needed. The transfer RNA is going to bring the bases plus amino acids. And then you're going to get these bases plus the new amino acids are going to give you your proteins that these codons were coding for. So you've got your transcription where it's coded onto the messenger RNA, and then you've got translation where it's decoded and the transfer RNA brings in what you need plus amino acids and your proteins are created. So that's kind of a complicated process, but it's what genes do. That's their job. Their job is to send messages throughout the cell so that new proteins can be created. And they are going to carry your hereditary information, and they have the basic instructions necessary to tell your body how to carry out the hereditary processes and keep producing the genes that keep producing the cells and the proteins that you need and maintaining your cells. So just keep in mind, genes are units of hereditary information, and they carry messages throughout the cell and instruct the cell on how to create the proteins that your body needs. Meiosis. Sexual reproduction in eukaryotes involves a form of cell division known as meiosis. It has several different stages that are carried out to let one diploid germ cell result in 4 haploid gametes. And diploid means that it's going to have multiple copies of the genetic information, where haploid means it's going to have one copy. So, in this first part, this would be interphase, and it's kind of before meiosis starts, but it has to do with it because this diploid germ cell contains genetic information from both a maternal and paternal parents. So you've got female and male genetic material in this one diploid germ cell, and it's getting ready to divide to carry out sexual reproduction. So we have replication going on in here because each chromosome is going to double itself so that there's extra genetic material to carry out this process. In the next phase, we have prophase, and the replicated genetic material is going to line up in homologous pairs, which means that each one is the same genetic material. It's an exact copy, and they're going to cross over a little bit at these points here, and where those cross over, they kind of swap a little bit of genetic material. So each one of these pairs is going to take a little bit of the other pair, and that is called crossing over or recombination. So you're just kind of jumbling up the maternal and paternal bits before the division starts to take place. Then we move on to metaphase. And in metaphase, your centrosomes are starting to pull the homologous pairs apart. And then we move on to anaphase. And You can see they actually are getting pulled further apart. They're not crossing over anymore, and they're starting to move to opposite sides of the cell. And these are all prophase 1, metaphase 1, anaphase 1, because this is kind of a two-step process. Then we keep going, and we've got telophase 1. In telophase 1, there's the cleavage furrow that's preparing for the cells to separate and become two distinct separate cells. There is a nucleus being reformed. The microtubules that have been connecting the chromosomes and pulling them apart have receded, and the genetic material is encased in a nucleus again. And then this last part is cytokinesis 1, where the cytoplasm is split, and there is a definite split made between those two cells. So now there are two diploid daughter cells. So we had our diploid germ cell. The DNA, the genetic material was all replicated, so there was twice as much. We went through this cell division, so now each one of these daughter cells is a diploid daughter cell, and it has twice as much genetic material as it needs to be a haploid gamete. So we're going to go through what is known as meiosis 2, the second step in this process. So we have prophase 2. And in this phase, you'll see we've got the two daughter cells, and now they're kind of repeating the same process that happened up top. So there's no replication because that was an interphase, but they are going to start having the centrosomes move down to either side and start moving out to opposite poles of the cell and start pulling the chromosomes that way. In the next phase, we've got metaphase 2. You can see that the chromosomes have lined up along the equatorial plate in both of these daughter cells. The microtubules are connecting them to the centrosomes, and they're going to start pulling apart. So in our next phase, anaphase 2, you can see that actually happen. The chromosome pairs have gotten pulled apart, and now these individual chromatids are on separate sides of the cell, and they're still going to be kind of being reeled in by the microtubules at this point. And we move on to telophase 2, where we've got a nucleus forming around our new pairs of genetic material that have both the mother and father genetic material in each nucleus. They're all split up now, and we have the cleavage furrow again. The cells are getting ready to divide permanently. Your centrosomes have separated to the opposite sides still, and the microtubules have retracted. And then we go to cytokinesis 2, which is where the cytoplasm divided between these two new cells and these two new cells, and now we've got 4 haploid gametes, which means that they each just have one set of genetic information, and they're going to need a partner to be able to reproduce. So these gametes are going to be ready to be fertilized, and when they are fertilized, a diploid zygote will be formed. So these gametes are ready to be fertilized, and if they are, then they can go on to create a new eukaryotic organism. So meiosis does have a 2-part process, and it goes through the same phases as mitosis, but they're a little bit different because instead of just getting an exact copy, you're mixing genetic material from 2 different organisms, a maternal and paternal parent, and at the end you're going to have the 4 haploid gametes that each has genetic information from both parents. Mitosis. Eukaryotic cells reproduce asexually by dividing into two genetically identical daughter cells. Now they do this by the process of mitosis. So let's look. This first phase is called interphase, and it's not really considered a part of mitosis, but it's usually discussed with mitosis because it happens at the beginning, right before mitosis happens. It's the build-up to mitosis. It's a period of cell growth and DNA replication. So the cell gets a little bigger, the nucleus gets a little bigger, the DNA replicates, so there's more of it for the mitosis that's about to happen. Chromosomes are in the form of uncondensed chromatin. And the nucleolus may still be present. So this would be our uncondensed chromatin. This darker dot here would be your nucleolus that you could still see. And these little pieces here would be your centrosomes. So whenever you see them along the rest of mitosis, remember these are the centrosomes. Okay, first stage of mitosis is going to be prophase. In prophase, the chromatin condenses into discrete chromosomes. So remember, it had gotten, we had uncondensed chromatin that the DNA had been turned into. In prophase, it condenses into discrete chromosomes. So instead of a bunch of DNA squiggled up, you've got discrete chromosomes that kind of look like little squiggly X's. The nuclear membrane breaks down. So where we had a complete nuclear membrane here, it starts to break down a little bit. And sister chromatids are joined by centromeres. So what they mean by that is this would be your chromosome, and these sister chromosomes or chromatids are joined by these centromeres. So this would be your centromere. And then each of these would be sister chromatids that form the chromosome. The centrosomes are going to move to opposite poles of the cell. So remember, we just kind of had them in the cell here. Now they've moved to opposite sides of the cell, so they're opposite each other and they're kind of equally spaced in the cell. The chromosomes have paired up to form sister chromatids connected by a centromere. And now we're ready to move on to our next phase, which is metaphase. In metaphase, the chromosomes align at the equatorial plate. So where they're kind of just all over the place, they've lined up at the equatorial plate, which means that it's equally distanced from both of those centrosomes that have spaced themselves out at either pole. The microtubules attach sister chromosomes to the centrosomes. So you've got your centrosomes, and these are your microtubules that are attaching the chromosomes to the centrosomes. They've lined them all up along this equatorial plate. And now we're ready to move on to the next stage of mitosis, anaphase. In anaphase, the centromeres divide. So remember, your centromere was that little center here that was kind of holding the two chromatids together. So in anaphase, they divide. The microtubules shorten, so where they're longer, they shorten up, moving paired chromosomes to opposite sides of the cell. So we had these paired chromosomes, and now the microtubules are shortening. They're moving those individual chromosomes over closer to the centrosomes now. And then you're ready for the last phase of mitosis, which is telophase. In telophase, the cytoplasm divides along a cleavage furrow. And so you can see how it starts to kind of dip in. It cleaves in half, and so there's that cleavage furrow, and that's going to show you where your cytoplasm is going to eventually split completely off and form two new cells. The chromosomes condense to chromatin. Remember, that's what we had up here. We had all the squigglies. They're going to just start condensing to chromatin, not a discrete chromosome anymore. And the nucleolus reappears, and the nuclear membrane is going to be reforming around a clear nucleus. So you see the nuclear membrane is forming around where your condensing or your uncondensing chromosomes are, and your nucleolus has reappeared. So once these cells split off, then this is going to be a nucleus again that looks much like this, with the uncondensed chromatin and the nucleolus in the center. Two daughter cells, as we discussed at the beginning, two identical daughter cells will be formed once cytokinesis is complete. And cytokinesis is that process of the cell being cleaved in half to form two new identical daughter cells. So this is mitosis. It is how a eukaryotic cell can reproduce asexually. It just divides into two identical daughter cells. It goes through interphase, prophase, metaphase, anaphase, and telophase. So it starts off with this uncondensed DNA, this uncondensed chromatin genetic material. The chromosomes form little pairs, and the centrosomes come to either side. The centrosomes attach to the centromeres with microtubules, and those chromosomes line up along the equatorial plate. Then they split apart, starting to look like two new cells now, and each part of the chromosome pair goes toward a centrosome so that they are split equally across the cell. And then they split apart, forming a new nucleus with a nucleolus, and cytokinesis is going to be complete once this cell finishes cleaving in half. So just remember that eukaryotic cells can reproduce asexually by mitosis. Photosynthesis. Photosynthesis is a process used by plants and other autotrophic organisms to convert light energy, usually from the sun, into chemical energy that can be used by the organism. Photosynthesis usually gives off oxygen as a waste product as well. Oxygen is not something that the plant or other autotrophic organism will need in most cases, and so it will give off oxygen as a waste product. Now, autotrophic means that these organisms make their own food. They are capable of creating their own food. They can harness the light energy and create their own source of nourishment. Heterotrophs cannot do that and must rely on autotrophs or other heterotrophs for their nourishment. Most plants, most algae, and cyanobacteria perform photosynthesis and are called photoautotrophs. So, autotrophs are organisms that can create their own food. Photoautotrophs are organisms that create their own food from light, since photo translates to light, looking at the Latin root for that word. These are organisms that can create their own nourishment from light. Photosynthesis maintains atmospheric oxygen levels and supplies most of the energy necessary for life on Earth. So as these autotrophic organisms take in carbon dioxide, they let out oxygen and help maintain the atmospheric oxygen levels, because people and other organisms exhale carbon dioxide, and the plants or other autotrophs take that carbon dioxide in and turn it back into oxygen, which we breathe. So if we didn't have photosynthesis, we'd be running low on oxygen. And so photosynthesis helps maintain our oxygen levels. It also supplies most of the energy necessary for life on Earth. Even though you don't feel like you're eating light or getting your energy just from light, if you eat a vegetable, then you're eating something that it has only been able to grow because it used light energy to provide nourishment for itself. So you are now eating that plant, which got its energy and was able to grow from light. If you were to eat a cow, if you had beef, if you had a hamburger, then you would be eating another heterotroph. Cows can't make their own food, but they ate grass and other vegetables and other foods that were grown, and they were grown by having light energy harnessed inside those plants. And so the cow got the energy from the plant, which got the energy from the light, and then you were getting energy from the cow. So it all comes back to the light energy. So while there are other sources of energy, the light energy is a primary source for everything going on Earth. Photosynthesis always begins when energy from light is absorbed by proteins that contain green chlorophyll pigments. So the autotrophs that perform photosynthesis don't always look green, but somewhere in their body, they're going to have these chlorophyll pigments that are able to take in and absorb light to start the process of photosynthesis. And photosynthesis occurs in two stages. You have your light reactions or light-dependent reactions. So these depend on light to happen. And it's when organisms capture and store the light energy. The light energy is converted to chemical energy in the form of NADPH and ATP. So in this light reaction or light-dependent reaction stage, the light energy is actually captured, turned into a chemical form of energy that can be stored or immediately used if necessary. In these carbon dioxide fixing reactions or light-independent reactions, these are ones that can happen even whenever light isn't present, as long as there was some energy harnessed in that plant already. So these don't have to happen while there is light present supplying energy. They can happen later. And this is when the formed NADPH and ATP, so it's already stored and formed in that autotroph, those two energy molecules drive the reduction of carbon dioxide to more useful organic compounds, such as glucose. And they are also referred to as the Calvin or Calvin-Benson cycle. So when the carbon dioxide is reduced, that process can be called the Calvin or Calvin-Benson cycle as well. So even though this is a light-independent reaction, it does still depend on molecules that were created by converting light energy into a stored chemical energy. So even though we call them light independent, they really couldn't occur if you didn't have the light transferring, the light energy that was able to be transferred to these energy molecules that are stored chemically. The main thing to remember about photosynthesis is that it is the process by which autotrophs can make their own energy using light. Proteins. Proteins are large biological molecules consisting of one or more chains of amino acids. Which are known as polypeptides. So a polypeptide is an amino acid chain, not just an amino acid, but the whole chain of them connected. Now, proteins have different structures based on how many of them are present. So the primary structure of a protein is its amino acid sequence. So just the one chain, the one polypeptide chain, and what sequence those amino acids are taking. The secondary structure is the regularly repeating local structures stabilized by hydrogen bonds. Now this is whenever we've got a few proteins interacting together and the local structures or the regular structures that these proteins will form just all on their own without any help. And some examples would be the alpha helix, beta sheet, and turns. So those are just some different shapes that the proteins can take whenever there are a few of them joining together with hydrogen bonds. Now the tertiary structure is getting more complex. It's the overall shape of a single protein molecule. So enough proteins have joined together to make a whole molecule, and the tertiary structure is referring to the spatial relationship of secondary structures to one another. So you've got your alpha helices, your beta sheets, and your turns, and it's how they interact together and what kind of shape they all form whenever they form into a single molecule. And you've got proteins joining together, and then more proteins with hydrogen bonds joining together into a larger molecule, and what that molecule looks like spatially. The quaternary structure is going to be the structure formed by several protein molecules. So you can see how we just keep adding proteins here to get different structures, and the more that are added, the more different that these structures are going to look because they're getting more complex. So several protein molecules are joined together in the quaternary structure, and this is usually called a protein subunit. So it's no longer one molecule, it's multiple molecules. So it's a subunit which functions as a protein complex. So it's going to be more complex, it's going to be able to process more complex functions. And a lot of the time what you're going to see is the quaternary structure of a protein, because you have lots of them together and they are drawn toward each other and then they bond. Now let's look at some of the functions of proteins. We have structural proteins, and these give stiffness and rigidity to biological components that would otherwise be more fluid. They wouldn't have a lot of shape to them. An example is keratin. Keratin is a protein found in our hair, nails, in birds' feathers, and in animals' hooves, and it gives them a hardness that they wouldn't otherwise have. It's not a part of your body like your skin or an organ, and you can feel they're a lot harder than your skin would be. Like your nail and your hair don't feel the same as your skin feels or as your internal organs would feel, and that's because they have this protein present that gives them more rigidity. So structural proteins are going to make things stiffer and give them a harder shape. Next we have enzymes. And enzymes catalyze chemical reactions. So there's a chemical reaction that needs to take place, but it won't be able to take place until this enzyme is present. When the enzyme is present, it catalyzes the chemical reaction and speeds it up and lets it occur. Next, you've got receptors. Proteins that function as receptors bind a signaling molecule to induce a biochemical response. So it receives that signaling molecule and then it binds it somewhere that will induce the biochemical response that needs to happen. Next, you have antibodies, which are also known as immunoglobulins, and these bind antigens and target them for destruction. So an antigen is a foreign body that comes into the cell and it's not welcome, it's seen as the threat, and so these antibodies or immunoglobulins are part of the immune system, and they bind to the antigens or that foreign body and they target them for destruction so that then they are destroyed and removed from the cell and the threat is eliminated. We also have motor proteins, and motor proteins generate the forces responsible for muscle contraction. So think about your motor functions, being able to move your muscles, being able to move around. All of those are are able to happen because of these proteins, these motor proteins. So if you're able to move your arm back and forth, if you're able to close and open your hand, those are those are happening because of muscle contractions, and those muscle contractions are generated by motor proteins. If we didn't have those, we wouldn't be able to contract our muscles like we are. Next we have pump proteins. So these are proteins that act as a kind of pump, and they transport ions or small molecules across a membrane. So you've got a cell membrane or an intercellular membrane, and the proteins can act as a pump and push these small ions or molecules across the membrane. And that is their primary function as a pump protein, is just to transport these small molecules or ions across the membrane. Lastly, we've got switch proteins, and these act as an on-off switch based on the presence or absence of certain other molecules in the cell. So if a cell was waiting for a certain molecule to be present to do something, then the switch protein is going to act as an off switch and keep the cell from performing that function until the certain molecule it's waiting on is present. Once it's present, it flips the switch and says, basically, it's on. It can perform this function now. So it acts as like an on-off switch for the cell. As you can see, proteins are very important in a cell. They actually can make up a large percentage of the components of a cell. For instance, you could have a cell that was made up of maybe 3% DNA, which is really important. You have to have the DNA encoded correctly for everything else in the cell to function. But the protein could make up almost 50% of that cell's components. So while it isn't maybe as biologically important as that DNA is, it makes up a big part of that cell, and not much would be going on if it didn't have protein there as well, because protein has a lot of functions. So you can see that proteins are very important for cell life and lots of cell functions. Punnett Square. A Punnett Square is a diagram of every possible combination of a maternal allele with its paternal counterpart for each gene investigated in a crossbreeding experiment. So that means if you were going to try to crossbreed two plants, say one that had red blooms and one that had white blooms, you could do a Punnett Square to predict how many different plants would come out red and how many would come out white and how many might come out pink. And this just would put out all the options, and from that you could figure out the ratios, and if you planted 100 of these plants or crossbred 100 of these plants, you could predict how many of them would come out which color, if color was the trait you were looking at. So let's look at some examples. A monohybrid cross considers only one trait in the cross. So mono means one, so we're only going to be considering one trait in the cross. So we're going to look at a tall maternal pea plant and a short paternal pea plant. So we're looking at peas, and the big T is going to represent the dominant allele, and the lowercase t is going to represent the recessive allele. So since the big T is dominant, anytime the big T is present, it's going to take precedence. So the big T means tall. So since we have two big T's, we know this is going to be a tall plant. If we see a big T and a little t, it's still going to be a tall plant because a big T is present. The only way we get a short plant is with two little t's because it is recessive, and so you must have both alleles be short alleles to get a short pea plant. So let's look at how this looks in the Punnett Square. So what we're looking at is a tall parent that was dominant and a short parent that had both alleles recessive. So let's combine them. We're going to put the big T with the little t in this box, big T with little t in this box, big T with little t in this box, and big T with little t in this box. So since we have 4 that are a combination of a big T and little t, this would give you a phenotypic ratio of 4. We only have tall plants in this combination because every time a dominant allele will be present. The phenotypic ratio means what the plant's going to look like. So this will be tall, 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 tall. It wouldn't matter if we had a pair of alleles that were both dominant, or if we had a pair like this where there's a dominant and a recessive. It would still be tall, and so that's all we're looking at is what it looks like, not what the alleles actually, what the letters of our alleles are in the Punnett square. So since they're all going to look tall, our phenotypic ratio is 4. They will all 4 be tall. So let's look at a different one, and I'm going to kind of draw us a line to keep us separated. And this one's going to be a dihybrid cross. And a dihybrid cross considers 2 traits in the cross. Di means 2, mono means 1. So we've got our dihybrid cross that's going to compare 2 different traits. So we're going to look at a tall wrinkled parent and a tall round parent. So again, the T's are going to be the same. If we have 2 big T's, it'll be tall. A big T and a little t, tall. 2 little t's will be short. Your R's are going to stand for whether it's round or wrinkled. So the big R is the dominant allele, and it codes for round P's. Round P's are the norm. Wrinkled P's are going to be that recessive gene. So a big R and a little r is still going to give you round P's because it includes a dominant allele. And the 2 little r's would give you the wrinkled P's because that is the recessive allele. So if all you have are the 2 recessives, you're going to get those wrinkled P's. So this is a parent that has a dominant and a recessive allele for tall and 2 dominant alleles for round P's, and another parent that has a dominant and recessive gene for tall and 2 recessive genes for wrinkled P's. And we're going to look at what we get in our Punnett square with this. So we have big T, big R, little r. So this is going to give us tall and round because we have 2 dominants and a dominant and a recessive. Moving across, we have 2 big T's, a big R, and a little r. So again, we're going to get tall and round. Next, we've got big T, little t, big T, or big R, little r. So again, it still includes 2 dominant, so we're going to have tall and round. Big T, big R, little r. We still have a dominant T and a dominant R, so it's going to be tall and round. Okay, moving on. Big T, big R, little r. Tall, round. And if you notice, these 2 are the exact same. So what you're going to put all the way across is going to be the same as what you had above it. Since we had a big T, big R, big T, big R, so the results going across the board are going to be the same. So all of these will also be tall and round. And if you wanted to go across and check them all, you definitely could, and just look at it. Big T with this little t, we've got it. Big R with this little r, we've got it. Okay, now we've got a different one. Little t, big R. So now we're going to have a big T with a little t. Big T, little t. And you always want to put the dominant gene or the dominant allele first whenever you're writing it. Then you've got big R, little r. So this is still going to code for tall and round because you have a dominant allele for the tall and a dominant allele for round. Next, you're going to have big T, big R, little r. So again, tall and round. Unless we have two recessives, we're going to continue to have tall and round. Okay, now we've got little t, big R, little r. So now we have two recessive genes. So we're going to have a short plant, but it's still going to be round because we have that big R. Okay, the next one, little t, big R, little r. And so again, we're going to have a short, round pea plant. Okay, this is going to be the same way that we did the top row. Since this was a little t, big R, and this is a little t, big R, the results are going to be the same across the board. Big T, big R, little r. And then we get to our little t, big R, little r for these last two because we've got the little t, big R, little r for both of those. And they're going to code the same as the ones above them. Tall and round, tall and round, tall and short. Wait. Sorry. Short and still round, and then short and round. So now we want to do our phenotypic ratio. We want to say how many of them look the same. So we know we have a lot that are tall and round, so let's count and see how many we've got. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we've got 12. The majority of our plants are going to be tall with round peas. And then we have 1, 2, 3, 4 that are short and round. So our ratio would be 12 to 4, our phenotypic ratio. That just tells us what the plants look like. Because if we were looking at the alleles themselves, some of the tall plants have two dominants for T, and some of them have a dominant and a recessive for T. But either way, they came out tall. So since they look tall, they get counted in the phenotypic ratio. Punnett squares can be kind of fun, and they kind of are like what you would do whenever you're graphing in math. So you just kind of have to make sure that you put every possibility out there. You put every allele along each side and then make sure you fill it in correctly and pay attention to how the alleles are going to code. Do you have two dominants coding for a dominant and recessive coding for a dominant, or do you have those two recessives coding for a recessive gene? You want to look at every possible option so that you can make an accurate prediction with your Punnett square. RNA, or ribonucleic acid, is made up of a base pair connected by sugar molecules and phosphate molecules. Now the bases in RNA are going to be adenine, uracil, guanine, and cytosine. Similar to DNA, they're going to pair with each other, and each one is only going to pair with one other base. They can't pair with any of the other three. So adenine pairs with uracil, and guanine pairs with cytosine. Guanine and cytosine also pair in DNA. Your curved letters for abbreviation pair together, and adenine and uracil pair together in RNA, and it's your vowels that are pairing together. Now in DNA, you have thymine as your other base, the base that pairs with adenine. But in RNA, you've got a different base. So you can think of it as your two abbreviations that are vowels paired together and your two curved abbreviations paired together. So adenine with uracil and guanine with cytosine. Usually RNA is going to be single-stranded. But it can form a double helix, like the shape of DNA, by folding in on itself. So you have a single strand made up of sugar molecules and phosphate molecules, and then you've got the little base pairs coming off of it. So it may be like this. You've got your sugar and phosphate molecules, and then you've got your little base pairs coming off of it. And my little lines here are splitting it. So you would have adenine and uracil, guanine and cytosine, adenine and uracil, guanine and cytosine, all the way down. Now, if the RNA was more complex, it may fold in on itself, and this could curve around and kind of form a double helix by being tightly coiled and wrapped together, so that instead of having just one side of the ladder, it curved up and made the one side kind of sandwich it together and form into a double helix that way. But it's not going to be the same kind of pattern, the same structure as DNA is going to be. RNA is also a carrier of genetic material in some viruses. So in viruses, RNA can carry the genetic material, and a lot of times if it's a more complex virus, you'll have RNA that is a double helix. It has formed a more complex structure to carry genetic material for the viruses. In most cells, DNA is what's holding all of the genetic material, but RNA is what kind of moves it around the cell. So let's look at the three main types of RNA. Messenger RNA, or mRNA, carries codes from DNA in the nucleus to ribosomes in the cytoplasm. So it's moving, it's being a messenger and carrying that code from the DNA to ribosomes. Ribosomal RNA, or rRNA, they read that code carried by the messenger RNA. And ribosomes are composed of the ribosomal RNA and proteins. So the ribosomal RNA is a big part of ribosomes. And then transfer RNA, or tRNA, bring amino acids to the ribosomes where they are linked to proteins. So you can see RNA is moving around the cell a lot. It's not just staying in one place. DNA is mainly just going to stay in the nucleus and hold that genetic material. RNA is going to move it around, except in the case of viruses, where RNA is the main holder of genetic material instead of just a transport system. So let me show you what this might look like. So let's say we've got our ribosome, and so you've got RNA, your ribosomal RNA in here. Well, you've got DNA in the nucleus over here, and it's going to get transferred to RNA via the messenger RNA. So it's going to send that code along, and it's going to come into the ribosomal RNA. And then you've got transfer RNA out here, and it's going to bring amino acids into the ribosome. So you've got your ribosome here, and your ribosome holds the RNA. It takes in the mRNA, reads the code from DNA, it takes in the transfer RNA and picks up these amino acids, and out comes a protein. So the whole point of all this was to create a new protein, and RNA is a big part of protein synthesis. So it's going to move the code from the DNA via messenger RNA into a ribosome, and transfer RNA is going to bring amino acids into the ribosome, and the ribosomal RNA is going to take the code and take the amino acid and create a protein and spit it out. So that's what's going on here. Remember the big differences between RNA and DNA? DNA is holding all that information in the nucleus. RNA is moving the information around. It's how DNA gets its code from one place to another. So RNA is more of a transfer system within the cell, and it has several different kinds of RNA so that it can do different jobs in the cell, all working on transferring that genetic code.
Catalysts are substances that speed up the rate of a chemical reaction without being consumed in the process. And so the way a catalyst works is first by lowering activation energy. And it lowers the activation energy that is required for a chemical reaction to proceed. And so by lowering that activation energy, the chemical reaction can proceed more easily. Now, another way that a catalyst meets its objective is by providing a surface for molecules to come together and to bind. And so to provide a surface for molecules to come together, to bind, is faster than just random collisions of reactant molecules. So that is how a catalyst meets its goal of speeding up the rate of a chemical reaction. Now, we can classify catalyst as homogeneous catalyst and heterogeneous catalyst. So a homogeneous catalyst is in the same phase as the reactants. So say you have a liquid reactant and you have a liquid catalyst, then it's a homogeneous catalyst. Now, a heterogeneous catalyst is just the opposite of that. The catalyst is in a different phase than the reactants are. So some examples of this would be, take liquid bromine, for example, and it speeds up the breakdown of liquid hydrogen peroxide into liquid water and oxygen gas. So you have liquid bromine and you have liquid hydrogen peroxide. So those are in the same phase, so that's a homogeneous catalyst. Now, take, for example, the combination of ethylene and hydrogen gases to make ethane gas. It's catalyzed by adding powdered nickel. So you have powdered nickel, which is in solid form, and everything else is in gas form, or the gas phase. So in that reaction, nickel is a heterogeneous catalyst. So an important point to remember is that catalysts are substances that speed up the rate of a chemical reaction without being consumed in the process.
Today we want to go over just some of the basics of chemical reactions. We don't have time today to do this thoroughly or fully in depth, but just want to go over some of the basics of chemical reactions. So when we think about chemical reactions, one of the things we ought to think about is time scale. How rapidly or slowly do chemical reactions take place? And in terms of time scale, obviously we're measuring this from our vantage point, from our viewpoint. So on the human time scale, the way we experience time, chemical reactions can happen very rapidly, very quickly, and also very slowly. From the lower end, just fractions of a second for a chemical reaction to take place, all the way up to the upper end, years and years and years and years and years before a chemical reaction fully takes place, and everything in between. So with chemical reactions, the time scale is from fractions of a second all the way up to lots of years for them to take place and everything in between. When we think about time scale, we also think reaction rates. What affects reaction rates? So in a chemical reaction, we need to think in terms of the frequency of the contact between the chemical components that are reacting. If it is very little contact, you have a slower rate. If they don't come into contact very often, that also slows it. If there's a great mixing of these things that react volatilely, you can have a very rapid reaction. So it's the frequency of contact between the interacting chemicals. The temperature plays a role. Higher temperature, lower temperature can affect not only the reaction but the time rate as well. And then the properties of the chemical that is interacting. Is it a solid, a liquid, or a gas? The shape it's in. All of these things play a part in reaction rates. Frequency, temperature, and properties. Then when we think about reaction rates, you can actually influence those rates. You can accelerate a reaction rate. This is called using a catalyst. A catalyst is introduced into that chemical reaction, which greatly speeds it up. And that's the catalyst's only job, is to come into this interaction between these chemicals and to speed up that reaction that would ordinarily take place over a slower period of time. You rapidly increase it. But you can also decrease reaction rates, and this is called using inhibitors. If you introduce an inhibitor into a chemical reaction, it slows it down. It keeps them from reacting as quickly. So you can affect reaction rates not only naturally and through ways that you're able to control temperature and what form it's in, things like this, but you can also introduce other chemicals, catalysts to accelerate and inhibitors to decelerate the reaction times of the chemical reaction. Now, chemical reactions can, when they occur, release heat, release light, electrons, ions. There's usually some form of product from this interaction, this chemical reaction that's taken place. Sometimes chemical reactions send the temperature in the opposite direction, but usually it's things like heat and light, electrons being traded or spun off, ions, radicals going off, things like this. And then finally, heat and other factors can influence chemical reactions and help break bonds. If you think in terms of breaking down carbon bonds, this is done obviously in the production of oil and related materials. You've got strong, stable carbon bonds that need to be broken down and split up. Heat is usually a thing that is primarily used to help break those bonds, divide those things up, and then sort them out to their various functions and purposes. And if you ever do any sort of research or study, it's very fascinating to look at an oil or a chemical plant and how they bring in the raw materials using heat and other pressure and things like this to break up the raw material that's brought in into various other materials, sifted out and sent other places and put to other uses. It's fascinating. But anyway, chemical reactions then, just the basic overview again, time scale from fractions of a second to years and years and years. Reaction rates are affected by the frequency of contact, the temperature at the point of their mixing and their contact, and then the properties, solid, liquid, gas, the shape of it, these sorts of things. Reaction rates can be accelerated with catalysts, decreased with inhibitors. Chemical reactions produce things often like heat, light, spinning off electrons, ions, things like this, and heat and other factors influence chemical reactions and help to break bonds. Once again, this has just been the basic overview of chemical reactions. If you'd like to learn more about this or related matters, underneath this video you'll find a link. If you'll click on that link, it'll take you to the website that has that information, and while you're at that website, you'll also find a link to an ebook that's ready for immediate download. I just want to take a few minutes now to go over briefly the 7 main functions of the integumentary system. 7 main functions of the integumentary system. One, it helps regulate body temperature through excreting sweat, controlling blood flow. So through sweating and blood flow, the integumentary system helps regulate the temperature of the body. 2, it provides protection, barrier to microbes, a barrier to UV radiation, to dehydration, protection from abrasions and from chemicals. Third, the integumentary system provides sensation, pain, temperature, touch. It's the primary means through which those sensations come to us, those type of sensations. It also helps with excreting sweat, water, or heat from the body. It helps with immunity. It produces antibodies and helps remove microbes by epidermal cells loss. So as the cells are lost, then surface microbes are taken with those cells that are removed from the body that way. It also provides a blood reservoir, a way to move blood to the tissues and organs and muscles as necessary. And then finally, it serves as a source of vitamin D production through UV radiation exposure. So the integumentary system's 7 main functions, once again, are regulating body temperature, providing protection, providing sensation, pain, temperature, touch, excretion, certain types of immunity, helping our immune system, blood reservoir, and then the production of vitamin D. This has just been a basic overview. If you'd like to learn more about this and related matters, underneath this video you'll see a link, and if you'll click on that link, it'll take you to a website with that information. And while you're at that website, you should also find a link to an ebook that's ready for immediate download. Today we're going to briefly go over an age-old philosophical question: nature versus nurture, or in other words, genetics versus environment. Are people born a certain way, or are they shaped and become a certain way based on the environment around them? Is it their nature? Are they born with it? Is it their genetics? Or is it nurture, their environment? And what we see is we look at the vast array and complex of human beings, that some things are almost 100% genetic, and some things are almost 100% environment, and some things are a clear combination of the two. So sometimes it's not an either or, it's a both and. And we're just going to briefly go over some of those things related to human beings: genetics versus environment, nature versus nurture, and then the combination of the both. So when we begin then with looking at genetics, the nature side, what we are born with, what we find as scientists study this is that things like the color of our eyes, our blood type, the quality of our vision, whether we need to have it augmented or repaired or helped through glasses and things like that, our teeth, certain diseases, not all, but certain diseases that we might be prone to, heart disease, things like this. There's been a clear genetic link found that children tend to have eye color based on what they've received from their parents. Whether it's a dominant trait or a recessive trait, the information has been passed down genetically. You don't literally change someone's eye color by anything in the environment, normally speaking, unless there's some sort of disease involved or some sort of purposeful augmentation through colored contacts or who knows what. But genetically speaking, our eye color is passed down to us biologically and has nothing to do with our environment. The same with our blood type, the quality of vision we have. Although the environment can affect our vision, whether it gets better or worse based on certain healthcare regulations, based on where the person is born, can affect it adversely one way or the other. But generally speaking, whether or not you're going to need glasses is not determined by your environment, but more by genetics. So there are certain things related to our bodies which we are just born with, certain proclivities to certain diseases, teeth, vision, blood type, eye color, things like that are clearly coded in our DNA and in our genes and passed down. Now there are certain things that are environmental and have nothing to do with our genetics. It's where we grow up, who we grow up around, that influence us. So things like language. The language you speak is shaped entirely by what you hear as you're growing up. I have a friend who grew up in a bilingual home. His parents were, one was of Japanese descent, one was of American descent. He grew up hearing both Japanese and English spoken to him as a child. Now genetically, he wasn't programmed to know Japanese or programmed to know English. He was just genetically programmed to receive whatever language came in. Since he received both, as he grew up, for a time there, he actually meshed both languages, and others around him often had a hard time understanding what he was saying because he was freely associating and mixing both Japanese and English. Now, eventually, as he got older, he was able to separate the two languages and speak them both fluently. But the fact is, it was the environmental influence of hearing both languages that gave him that capability. So the language that you are exposed to from the time you're born growing up is, generally speaking, the language that you will speak naturally. You can also then learn other languages through school and classroom or living in another country. You pick up the language. So language is one of those things that is, a specific language is one of those things that's environmental and not genetic. Now our ability to receive, comprehend, assimilate, process, and use language is something that's genetically inherent in us. There's some sort of matrice or lattice in the brain that allows the reception and processing and then use of a language. But what language we speak has nothing to do with our genes. If our children were born in Japan, they wouldn't be born speaking Japanese because they were born in that country, or they wouldn't be born automatically speaking English just because their parents spoke English. It's something that their environment would program into them. Along with that are things like religion and other social traits. What you grow up around socially tends to influence things like that in a person's life. So people aren't born a particular religion genetically from their parents just because their parents are. It's something that is socially around them and that they pick up. Same with other social traits, whether they're feel like they are motivated by guilt or by shame. We have different cultures that are guilt-based or shame-based. It's not something that's genetic, but something that's environmental and social. So those factors get a little more complicated, but it's clear, it's obvious. We picked examples to show that which is clearly genetic, that which is clearly environmental. Now there are some things that are actually a mix of the two, a conflation of the two. Things like our height. Now you might say, well, height would automatically fall under genetics. It falls partially under genetics. How tall you are is partially genetic. But environmental factors actually play a role in that. If your diet is not healthy, if it's suppressed, if it's missing necessary nutrients or enough protein, you may not reach your full height genetic potential. Your growth may be stunted because of environmental malnutrition and things like this. So how tall you get is a mix of both genetics and environment. The same with your weight. You may be genetically predisposed to store more fat in your body in particular places. You know, we know apples and pears. Apples stored above the waistline, pears store it below the waistline. This may be a genetic thing in where you store fat and how much you're prone to store, but once again, environment plays a huge role. If you're malnourished, you won't store as much fat because you won't be able to. Once again, if you overeat or eat too much of certain kinds of food, you may store too much fat. So both genetics and environment play a role in things like height, weight, and then of course with diseases. You may have a genetic predisposition to a particular disease, but you can environmentally take steps to help prevent that through exercise and diet. You may also inadvertently not take steps to prevent that and walk right into it at an earlier age because environmentally you didn't take the steps necessary to help guard against something that's in your past genetically handed down to you. So we've gone over the whole nature-nurture debate here that's often argued in philosophy and realized that when research is done, when scientific observation is done, we find that some things are clearly genetic, some things are clearly environmental, and some things are clearly a combination of both to varying degrees. This has been just an overview of this very interesting and detailed topic. If you'd like to learn more about this or related matters, underneath this video there's a link. If you'll click on that link, it'll take you to a website that has that information, and while you're on that other website, you'll also find a link to an ebook that's ready for immediate download.
Metals can be found in various places on the periodic table. They are generally found on the left side. So if you're looking at the periodic table and you're looking for metals, you're going to find most of them on the left. So the first group of metals is in group 1A, which is the first column of the periodic table, all the way on the left. It's a pretty tall column, and it started off by hydrogen. Now, hydrogen is a non-metal, but all of the elements under it are metals. And so we call group 1A the alkali metals. And so some of the alkali metals include sodium, potassium, lithium, and cesium. And then we get to group 2A, which is the second column of the periodic table, the column 1 over from group 1A. And we call those the alkaline earth metals. So some metals in that group include calcium, strontium, magnesium, barium, and radium. And then we get to groups 3 through 12, which is a big chunk of the periodic table. And so you have some tall columns on the periodic table on the left, and then you have some tall columns on the right. And so then there's lots of shorter columns in the middle, and so that's what groups 3 through 12 is. And we call those the transitional elements. And so the transitional elements, or we can also call them the transitional metals, because all of those are metals in there. So there's a lot, but some metals in that family include manganese, nickel, iron, copper, gold, mercury, zinc, and silver. And then we get to groups 13 through 16, which is the columns all the way on the right. And you may be thinking, not all those elements are metals. And that's correct. The metals up near the top are non-metals, and the metals below it are the ones that are metals. And so it's only the elements that are below the metalloids that are actually metals. And so some periodic tables actually have a bold, jagged line. And all the elements below that bold line are the metals. So you can look for that on some periodic tables. And so that's a look at where you can find metals on the periodic table.
going to talk just a little bit today about methods for taking blood pressure. And when we think about methods of taking blood pressure, it breaks down into two categories: invasive and non-invasive. Invasive methods of taking blood pressure, non-invasive methods of taking blood pressure. Now, when we think about invasive ways of taking blood pressure, what we're talking about is inserting a cannula into a blood vessel and connecting that then to an electronic pressure transducer. So an electronic pressure transducer reading the blood pressure as it comes from the cannula inserted into the blood vessel. Now, where this is primarily done is in ICUs, anesthesiology, and research, where you need very accurate results. And so invasive methods of taking blood pressure are the most accurate means of obtaining blood pressure. Obviously, it's critical when you're an anesthesiologist dealing with a patient during a procedure, a surgery, things like this, in an ICU, critical situation there, and also for research where you need the results to be very accurate for the research. Complications, though, from invasive methods of blood pressure include infection, since you are inserting something from the outside into the interior of the body, directly into a blood vessel. Infections are possible, and of course, bleeding is possible. So it is more painful for the patient if they're awake while this is happening, because once again, it's inserted into the body. So things to remember on invasive methods of taking blood pressure: it's most accurate. Complications include infections and bleeding. Most common areas where an invasive method would be used would be an intensive care unit, anesthesiology, and research, and it involves inserting a cannula into a blood vessel and connecting that to an electronic pressure transducer. Then we'll conclude with the second category of methods for taking blood pressure, which are non-invasive methods. Primarily an arm cuff is what we're thinking about here, although there would be others. It's an external means, no invasion of the body whatsoever, an external means of monitoring blood pressure. Things to remember under non-invasive methods of taking blood pressure are that it is less accurate. It is less accurate. Not that much less accurate, but it is less accurate. It is also much less painful for the patient. Therefore, if it's not critical, like you'd find in one of these areas here, then obviously a non-invasive method would be best. It's more comfort for the patient, less painful for them, and the lack of accuracy in these contexts is not that critical. Places it would be used would be things like in a typical doctor's office when you're just trying to get the baseline basics of their stats, and blood pressure is included in that, and then also home monitoring of blood pressure. An external means is obviously indicated there, arm cuff monitor of some sort. The good news about non-invasive methods of taking blood pressure is that there are no complications. There is no risk of infection, no risk of bleeding or anything like that, as it's just merely wrapped around the arm or wherever for a brief period of time while the blood pressure is taken. So these are the things to keep in mind, the two categories for methods of taking blood pressure: invasive, non-invasive. Under invasive, remember most accurate. Remember that there are complications possible of infection and bleeding, and that it's used usually in areas such as intensive care, anesthesiology, and research. Non-invasive, less accurate, but not much so. Much less painful, no complications. Standard areas where it's used is doctor's office and home. If you'd like to know more about this and things related to it, underneath this video you'll see a link. Click on that link, it'll take you to the website with that information, and while you're there you'll also find another link to an ebook that's ready for immediate download. The brain is one of the most important parts of our nervous system, and today we're going to go over the various parts of the brain and talk about some of the ways they contribute and help in the areas over which that part of our brain control. And so we're going to begin then with the frontal lobe. The frontal lobe controls our emotions, our judgments, and our motor aspects of our speech, and is also the primary motor cortex for voluntary muscle activation. After the frontal lobe, we have the parietal lobe, which receives sensory information from touch. Proprioception, which has to do with our ability to tell where our arms and legs are and our orientation in space. So even with your eyes closed, you know where your arms and legs are. You have a sense of your body. This is proprioception. The parietal lobe helps govern that. Also our sensation of temperature and pain. That's the parietal lobe. Then the temporal lobe is responsible for our hearing, auditory information, and our language comprehension. The occipital lobe is our center for visual information, our sight. The cerebellum coordinates all of our muscle function, and then we have the brain stem, and within the brain stem we've got the midbrain, the pons, and the medulla. The brain stem governs our breathing, our respiration, our hearts, our cardiac center, and all the nerve pathways to the brain come through the brain stem. And then finally, moving on then from that to the diencephalon, which is the thalamus, subthalamus, and hypothalamus. The thalamus helps integrate and relay sensory information from the face, feelings there, our retinas, our cochlea from our ears, our taste receptors, and interprets sensation of touch, pain, and temperature. So our thalamus is very important to not only our sense of what we feel in terms of pain, temperature, touch, but also our retinas, our eyes, our cochlea, and our ears, and our taste and our tongue. The hypothalamus controls the autonomic nervous system and the neuroendocrine system. It maintains our body homeostasis, helps regulate body temperature, helps regulate our appetites, governs our thirst and how we feel that, our sleeping cycle, and hormone secretions. So once again, the brain, very integral, necessary, core part of our nervous system, broken up into these various regions and what they controlled. If you'd like to find out more about the brain and its relation to our nervous system, I encourage you to go to the link that you'll find below this video. At that website, you'll find an ebook that is ready for immediate download.
Potential and kinetic energy. Potential energy is the amount of energy an object has stored due to its position or orientation. So potential energy is how much energy an object has when it's sitting still. It's just stored. It is not in motion. It's how much energy an object could have if it moved. It's potential energy. And gravity most commonly affects potential energy. Now there are other factors such as mass that can affect potential energy, but the force of gravity on an object is going to affect it the most. So if an object is up higher, it's going to have more potential energy than an object that's down lower, because the lower object is going to have a smaller amount of space to fall. The higher object is going to have a further way to fall, and so it's going to have more potential energy. The formula for finding potential energy: PE stands for potential energy, equals mgh. M stands for mass. G stands for acceleration of gravity. So the force that gravity is going to have on that object. And H stands for height. So you'll multiply the mass of the object times the acceleration of gravity times the height from which it is from the ground. And that is going to give you your potential energy. So you can see that the acceleration of gravity and the height, the distance an object is from the ground, are both big parts in this formula for finding potential energy. So gravity is very important to the potential energy in an object. So potential energy is how much energy an object has stored while it's sitting still. Kinetic energy is the energy of an object in motion. So when an object is actually moving, that is whenever you're seeing kinetic energy. And kinetic energy, Ke, can be found with this formula: mv squared divided by 2, where m equals the mass and v equals velocity. So mass times the velocity squared divided by 2 will give you the kinetic energy of an object. So this one doesn't have as much to do with gravity, because once it's in motion, the kinetic energy has more to do with the velocity and the mass of the object. So let's look at this. So we've got this little situation set up. You've built this out of wood or Legos or something, and you've got a ball and you're going to roll it down here. So when your ball is here, you have potential energy. It's sitting still. Once it goes over the edge, and you have it here, you have kinetic energy because this ball is in motion. If it moves down some more and sits still for a minute, you're going to have potential energy as it's sitting still because it could still go down some more. And if it does, then you have kinetic energy again because this ball is rolling, it's in motion. And then if it lands here, you've got potential energy again because it can still drop off this ledge. So potential energy is energy that's stored while an object is still. Kinetic energy is the energy of motion. It's while something is actually moving. So potential is still and stored. Kinetic is moving in motion.
Water is essential to life as we know it, and it is one of the main constituent of many living things. And so there's several interesting properties of water I want to take a look at. So first is its high polarity. It has negative and positive ends. And this is due to the fact that water is made up of 2 hydrogen atoms and only 1 oxygen atom. Then also hydrogen bonding. Water molecules engage in hydrogen bonds with other water molecules. Water also is cohesive. And so cohesiveness means it's attracted to itself. You may have noticed this if you ever filled a cup of water up a little bit too high above the rim of the glass. Well, the water didn't immediately spill over because of cohesiveness. The water molecules were attracted to the other water molecules, so the water didn't necessarily spill out because the attraction was stronger than the force of gravity trying to pull it out. Then we also have adhesiveness, which means water molecules are attracted to other molecules. Water has a high specific heat, which means it resists the breaking of its hydrogen bonds and resists heat and motion. So specific heat means that it has a large amount of heat required to raise temperature by a specific amount. This also kind of goes along with high latent heat, which is the net heat change in change of state. So basically when water changes from one state to another, that means there was a lot of heat either absorbed or put off. And so net heat change means how much water was absorbed or exerted to do this change of state. Now, water in its solid form as ice floats in its liquid state of water. Now, this is pretty odd because many times substances are denser in their solid forms, but water is strange in that its solid state floats in its liquid state. This is because when water is frozen, it expands, and so it becomes less dense because it's taking up a larger amount of space. So that same material is spread out over a larger amount of space, which means it's less dense than the liquid water. And so when anything is less dense than water, then it's going to float in that water. So that's why ice cubes float. So that's a look at some of the interesting properties of water. In a chemical equation, it is necessary to have the same number of atoms for each element on each side of the equation. So right here, we have a salt. And so we have 3 numbers here. We have a big 2, smaller 2, and a smaller 4. And so this big 2 right here is called a coefficient. And these smaller numbers are referred to as subscripts. And so they serve different functions. So a coefficient tells how many molecules, and a subscript tells how many atoms. And so when you're trying to balance a chemical equation to make sure you have the same number of each type of atom on the left and right side of the equation, you cannot change the subscripts because the subscripts control how many atoms are in that molecule. And so that's the particular makeup of that molecule. For example, if we change this 2 right here to a 1 on sodium, it would totally change the molecule we're dealing with. But if we change this 2 all the way up to an 8, it wouldn't change the makeup of this molecule because this right here is just the moles of this molecule. It's how much of this molecule we have. And so it would just mean that we have 4 times as much of this molecule if that number changes. So right here, we have a great example of how to balance a chemical equation, because we have diatomic hydrogen combining with diatomic oxygen to form water, H2O. Now, the reactants are always on the left side of the equation, and the products are on the right side of the equation. So these things over here are reacting together to form this product or products. And so the reactants are always on the left, and so we need to count how many of each atom we have on this side. So we have a subscript of 2 here, so we have 2 hydrogens, and we have 2 oxygens. Now over here on the right, we have a subscript of 2, so we have 2 hydrogens, and we have 1 oxygen. Now this one doesn't have a subscript at all, but it's just an understood 1. So we could write a 1 right there. But just to make things easier, instead of writing a 1, it's left out many times. And so in order to balance this, we can't change the subscripts, because the easiest thing to do would just be to change that 1 to a 2. But we can't do that because we'd alter the chemical makeup. And so we have to alter the coefficients. So what we can do here is put a coefficient of 2 in front of water. So now we have 4 hydrogens over here, and we have 2 oxygens. So now the oxygens are equal, but the hydrogens are not, because now we have 4 hydrogens over here. So we can put a coefficient right here, and so we have 2 moles of this hydrogen molecule, and so now we have 4 hydrogens total. So we just multiply that coefficient by the subscript, and so that is equal as well. So that's how you balance a chemical equation. Ionic bonds tend to form under 2 certain criteria. And the first is ionic bonds tend to form between metals and nonmetals. So if you have 2 elements and one's a metal and one's a nonmetal, an ionic bond is likely to form. Now the second criteria is that ionic bonds tend to form between elements with a large difference in electronegativity. So the larger the difference in electronegativity, the better the chance ionic bonds are going to form. And so if elements meet both of these criteria, there's a very good chance they are very likely to form ionic bonds. Now, keep in mind that elements with the highest electronegativity values are in the upper right-hand corner of the periodic table of elements, while those with the lowest are near the lower left-hand corner. So you have elements with low electronegativity down here, and elements with high electronegativity up here. And so just by knowing those 2 facts right there, if you see an element on the periodic table, if you just imagine this board as the periodic table, and it's about right here, you just know by where it's geographically situated on the board that there's a really good chance that it has a high electronegativity because it's near the top right-hand corner of the periodic table. Now, I have some pairs of elements here, and I want to examine these elements to see if they meet this criteria and determine whether or not they are likely to form ionic bonds. So first we have nitrogen and oxygen. Now, nitrogen is a nonmetal, and then oxygen is obviously a nonmetal. So they are not likely to form ionic bonds because there is not between a metal and a nonmetal. We have 2 nonmetals here. Then we have potassium and fluorine, and potassium is an alkali metal, and fluorine is a nonmetal. So we've met the first criteria. We have reaction here between a metal and a nonmetal. So we're doing good so far. And then there's a high electronegativity difference here, and it's actually 3.2, which is pretty high. So they're likely to form ionic bonds, specifically potassium chloride. Now here we have barium and sulfur, and barium is a metal, and then sulfur is a nonmetal. So here we have a metal, a nonmetal. We're doing good so far, so they're likely to form ionic bonds, specifically barium sulfide. And then in addition to that, their electronegativity difference is moderately large at about 1.6. Now finally, we have cesium and tin. And cesium is an alkali metal. And then I'm sure you can guess that tin is a metal. So here we have two metals interacting with each other, so it's not between a metal and a nonmetal. So these cesium and tin is not likely to form ionic bonds. So notice here the main factor here was that we had two nonmetals and two metals here. But when we have a metal and a nonmetal, they're likely to form ionic bonds, and especially if they have a high electronegativity difference. So hopefully through this short session, you have a better understanding of ionic bonds. There are 5 major classes of hydrocarbons that we're going to take a look at. So we're going to start with alkanes. And so alkanes consist of only carbon-carbon bonds and carbon-hydrogen single bonds. So an example of that is methane, which looks like that, and then also ethane, which looks like that. Now a second class of hydrocarbons is alkenes, which have at least one carbon-carbon double bond. And so an example of that is ethene. And so see here the double bond between the two carbons. The third class is alkynes, which have at least one carbon-carbon triple bond. So we're building up here. So an example of that is ethyne. So notice the triple bond here between carbon and carbon. Now the fourth type is cyclic hydrocarbons. Now cyclic hydrocarbons are ringed structures with carbon-carbon bonds. Now the carbons are bound together in a triangle shape, and each carbon has 2 hydrogen atoms bonded to it, one above the plane of the triangle and the other below. And so again, that's called a cyclic hydrocarbon, and an example of that is this right here, which is cyclopropane. Now the fifth and final major class of hydrocarbon that we're going to take a look at is aromatic hydrocarbons. And so aromatic hydrocarbons are ring structures with carbon-carbon single bonds and carbon-carbon double bonds in an alternating pattern. So an example of that is benzene, which has 6 carbon atoms and 6 hydrogen atoms. Now benzene is an aromatic hydrocarbon and is a widely used organic solvent. In chemistry, we come across what we can call covalent bonds. And these covalent bonds are formed when one or more pairs of electrons are shared between atoms. So the key word here is shared. Now we can take that a step further, and we have something called nonpolar covalent bonds. So I have a couple of blanks to fill in on this definition. So 2 atoms with equal electronegativity that share the electron pairs equally. So that's what nonpolar covalent bonds are. 2 atoms with equal electronegativity that share the electron pairs equally. So the key words here are equal and equally. So they have to have an equal electronegativity, and that means they share these electrons equally. Now, the opposite of this would be polar covalent bonds, which don't share the electron pairs equally. In other words, the electrons spend more time with one atom over the other. So we have this general definition of covalent bonds, which just means they share atoms, but it doesn't describe how they share them. So we have nonpolar and polar. But the key definition here is nonpolar covalent bonds, who do share the electron pairs equally. Now there's one more definition I need to give you, and that's the definition of the octet rule, which is outermost shells must contain 8 electrons to be complete. So the key word there is 8. So say, for example, we have oxygen. And oxygen has 7 electrons. So we have 2 oxygens here, and these dots represent an electron, so they each have 7. But according to the octet rule, the outermost shells must contain 8 electrons to be complete. So what they can do here is become covalent bonds. Meaning that they're sharing electrons pairs equally. So all these electrons around the outside are their own electrons, but this electron pair right here in the middle is being shared equally between the 2 oxygens to become diatomic oxygen. Now there is an exception to the octet rule, and that is with hydrogen and helium. And so these only need 2 electrons. And so say we had hydrogen, it has 1 electron, we have hydrogen, another hydrogen with 1 electron. They can come together and they can share that electron pair equally between them, and now they have all the electrons that they need. Okay? And so the way nonpolar covalent bonds usually come about is when 2 nonmetals chemically bond together. And so what these atoms are trying to do is have a complete outer shell of 8 electrons. And so what we have here now is 2 hydrogen atoms, and over here we have 2 oxygen atoms. So we have diatomic hydrogen, diatomic oxygen. And so these are exhibiting nonpolar bonds because the atoms have equal electronegativities. So sometimes you may be dealing with 2 atoms that are of different elements. But here, since the atoms are the same element, you know they're going to have equal electronegativities because they're the same type of atom. And because they have those equal electronegativities, they're able to share those electron pairs equally as well. Every element has an atomic number, and that atomic number is the amount of electrons that an atom of that element has. Now, if you look at a periodic table, as you move to the right of each row, you'll see that the elements have higher and higher atomic numbers. And then when you go to the next row, you'll see the same thing. The atomic numbers keep getting higher and higher as you progress down the periodic table. Now, all of those electrons have to go into orbitals. And so we're going to take a look at the order of electron filling in the periodic table. So if you take a look at row 1, you'll see that it only has 2 elements, hydrogen and helium. Hydrogen has an atomic number of 1, meaning it has 1 electron, and helium has an atomic number of 2, meaning that it has 2 electrons. And so helium is going to fill the s orbital. And so we could write it like this, row 1, 1s, because it fills the s orbital on energy level 1. Now, the only orbital on energy level 1 is the s orbital, and every orbital is capable of holding 2 electrons. So the s orbital of energy level 1 is capable of holding all the electrons of helium. Now, when you get to row 2 and to row 3, things get a little bit more complicated. It fills the s orbitals, then the p orbitals. Now, electrons want to be in the lowest energy state. That's why they start filling up the orbitals in energy level 1. Then they progress to energy level 2. Now, when they get to energy level 2, the electrons are first going to fill up the s orbital because it has a lower energy level than the p orbitals. So if you were to look at the element argon, which has an atomic number of 18, it's all the way to the right of row 3. So out of rows 2 and 3, argon has the highest atomic number. So 18 electrons is a lot of electrons to deal with. So row 3 requires the use of the s orbitals all the way up to energy level 3, and the p orbitals also all the way up to energy level 3. Now, the p orbital does not come into existence until energy level 2. So the 3 s orbitals are capable of holding 6 electrons total. Now, there are 3 p orbitals at each energy level, and the p orbital is in existence at energy level 2 and energy level 3, so a total here, it can hold 12 electrons. And if you add those together, you get 18. And so argon is taken care of by using the s orbital all the way up to energy level 3 and the p orbital all the way up to energy level 3 as well. Now when we get to rows 4 and 5, we come across an element called xenon, which looks like that and has an atomic number of 54, which is a lot of electrons. And xenon is all the way to the right of row 5. So we could write row 5 like this: 5s, 5p, 4d. Now, the d orbital does not come into existence until energy level 3. And there are 5 d orbitals at each energy level it exists. So at each energy level, the d orbital can take care of 10 electrons. So at row 5, to take care of xenon, we're using the s orbital all the way up to energy level 5. So that's 10 electrons the s orbitals can take care of. And then the p orbital is in existence by energy level 5 for 4 total energy levels and can take care of 6 electrons at each level. So that's 24. And then the d orbital can take care of 10 electrons at each energy level. And it's been in existence since energy level 3, so it can take care of 10 electrons as well. Excuse me, it can take care of 20 electrons total between the two energy levels. So we had 10 plus 24 is 34, plus 20 is 54. So you can see how it can take care of all the electrons of xenon. Now, row 4 would be written like this: 4s, 4p, 3d. Now this presents a really easy way to remember this. At row 5, it fills up the energy levels of the s and p orbitals at the same number. See, it's row 5, 5th energy level, 5th energy level, and then when we get to d, it's just 4, the number before. Then when we get to row 4, or when we go back to row 4, you see that the s and p orbitals are filled up at the 4th energy level, and we're on row number 4, and then d again is at the previous energy level at number 3. And it works similarly at row 6 and 7. Like row 6 would be written like this: 6s, 6p, 5d, and 4f. And then row 7 would be written like this: 7s, 7p, 6d, 5f. So notice that the s and p orbitals are filled up at that energy level. So on row 6, it's going to be 6, 6, and then we go to the previous energy level, and then we go to 2 previous energy levels. Now you probably have not seen the f orbital before. That's because it started at the 4th energy level. And there are 7 f orbitals at each energy level. So that means at each energy level, the f orbitals can take up 14 electrons. So if you look at row 7 right here and all that it's capable of taking care of a lot of electrons. So the important thing to remember is that the electrons of any atom have to go into an orbital. And so there's a specific order of electron filling in the periodic table. And if you look at a periodic table, the atomic numbers get higher and higher as you move to the right of the periodic table. The periodic table is a way to systematically display the chemical elements. Now notice I said that it consists of chemical elements, and it consists of elements only. So this does not include mixtures and compounds, because an element is basically matter in its most basic form. The periodic table positions chemical elements based upon atomic number. An atomic number is the amount of protons an atom has in its nucleus. So hydrogen is the first element on the periodic table because it has 1 proton in its nucleus. And then the next chemical element has more protons in its nucleus. Like I said, the periodic table arranges chemical elements very systematically. So if you were to take a look at one row on the periodic table, all the chemical elements on that one row would have the same number of electron shells. And if you were to take a look at one column, all of the chemical elements in that column would have the same electron configuration. So the periodic table arranges the elements in such a way that all the elements are next to similar elements. So if a scientist knew a lot about one element, he could know a lot about the elements around it just because all the elements are grouped together with similar elements. There are about 118 elements out there, but only 114 have been officially recognized. And 98 of those are natural elements, ones that naturally occur in nature, such as potassium, nickel, and hydrogen. The other ones are synthetically produced by humans, such as Einsteinium. So the important thing to remember about the periodic table is that it consists of chemical elements and it is arranged by atomic number. pH is basically a measure of how many hydrogen ions are in a substance. pH is very important to many fields such as biology, chemistry, medicine, food science, farming, and water treatment. I've written up here on the board a pH scale ranging from numbers 0 to 14, each indicating the amount of pH in a substance. Now there are pHs that go higher than 14, but for practical purposes, this is the largest pH scale we need. Water has a pH of 7, and any substance with a pH larger than 7 is considered a base. So everything from 7 over to 14 is considered a base. Common bases that you probably know of are baking soda and bleach. Now any substance with a pH below 7 is considered an acid. Some of those that you may know of are lemon juice, tomato juice, and gastric acid. So water is kind of the middle bar on the pH scale, and anything to the right is the base, and anything to the left is an acid. The science process skills include observing, hypothesizing, ordering, categorizing, comparing, inferring, applying, and communicating. These are all important science skills, and for the most part, this list goes from easiest to hardest. So if you want to understand one science skill, it's good to understand the science skills before it. So I want to go over all of these and give a brief description of each. So observing is pretty self-explanatory. This is being able to write down an accurate description of something observed in nature or something observed in an experiment. And then hypothesizing is building off an observation. You take an observation and then you form a prediction based off of that observation. Now, ordering and categorizing are similar because they both have to do with the structure of information. In ordering, you're taking information and maybe putting it in a list from most important to least important, or you're just putting it in some kind of order to make the information very accessible and very understandable. And then categorizing has to do with taking the information and putting it with other like information so that you have different categories of information. Comparing has to do with noticing the similarities between different events or different experiments. This is important because sometimes two experiments or two scientific phenomena will look totally different, but through the skill of comparing, you'll be able to notice the similarities between the two. Now, inferring is different than hypothesizing because hypothesizing is making a prediction of something that will happen, whereas inferring is making a prediction or is making a conjecture about something that did happen. It's hypothesizing is looking forward to a future event, while inferring is looking back to a past event, attempting to explain it from the information already known. Now, applying is taking a prediction and putting it into an experiment. In other words, you're taking your observation and your hypothesis of what might happen and actually applying it to an experiment. And finally, you have communicating, which actually goes hand in hand with observing, because observing is making an accurate description of something you have seen. Well, communicating is taking those observations and being able to communicate or tell that to someone else. That may be by writing a description of it or by telling them about it. But this is very important because by being able to communicate information, scientists are able to feed off of each other's discoveries. So that's why it's important to be able to accurately communicate scientific information. So those are the science process skills. The three classical states of matter are solids, liquids, and gases. So first we're going to look at solids, and we're going to look at some of the characteristics of a solid. So solids have a definite volume and density. So we would say they have a definite volume and density at a given temperature and pressure. So like this whiteboard, for example, is a solid. So the volume of this whiteboard isn't changing, and the density of this whiteboard isn't changing. And then the second point of a solid is it has a degree of structural rigidity and a constant shape. So notice that this whiteboard has structural rigidity and a constant shape, so the shape isn't changing. It also has a resistance to flow. Now, some solids, such as modeling clay, can flow and undergo deformation under pressure. So I'm just going to write here, there are exceptions, but for the most part, solids have a resistance to flow. Now, the second state of matter is the liquid. And like solids, at a particular temperature and pressure, it has a definite volume and density. So the reason I say at a certain temperature and pressure is because at a different temperature and a different pressure, this whiteboard may have a little bit of a different volume and a little bit of a different density. But as long as it stays at the same temperature and pressure, then it's going to stay the same. The volume and density are going to stay the same. The same thing with a liquid. Now, a liquid does flow readily, so that makes it a lot different than a solid. But it does not expand to fill a container. So it's going to flow readily. If you spilled water on the ground, it wouldn't all just stack up in one spot. It would flow all over the place. Well, if you were to pour water into a glass, the water isn't going to expand to fill up that whole glass. It's going to keep the same volume. Now, that's different than the third state of matter, which is a gas, because the main thing about a gas is that it will expand to fill a container. So all of a sudden, if a gas was exposed in this room, it wouldn't all stay in one spot. It would eventually move to fill the entire room. Now, the molecules are spread much farther apart, and they move more rapidly and randomly than in a liquid. So basically, the molecules move the slowest in a solid, they move a little faster in a liquid, and then they move the fastest in a gas. And so that's why gases are spread apart, is because they're moving around so much and bumping into each other and pushing away from each other. And a liquid has more movability to it, I guess, if that's a word. It can move more because the molecules move more. And then a solid, you think about ice, it's not really doing anything. It's just sitting there because it's a solid. The molecules are moving really slowly. And so a gas is far more dispersed than even a liquid because it'll expand to fill an entire container. And so that's a look at the three states of matter. The scientific method is a process that has been around for over 1,000 years that is used by scientists to make scientific discoveries. There's a progression in the scientific method. The scientist starts by asking a question that the scientist wants to have answered. In this case, the question is going to be, which cat is faster? The scientist is interested in knowing if the lion or the cheetah is faster. Then the scientist, based off of prior knowledge, is going to formulate a hypothesis. In this case, the hypothesis is going to be the cheetah is faster. Notice how the hypothesis is written as an established fact. It's because through the experiment portion of the scientific method, the scientist is going to try to prove his hypothesis true. Then comes the prediction portion of the scientific method. In this portion, the scientist is going to try to clarify his hypothesis. So in this case, the prediction is going to be the cheetah will be faster over a 60-meter period. It's important to realize the difference between a hypothesis and a prediction. A hypothesis is stated as established fact, and the experiment is either going to prove or disprove the hypothesis. The prediction clarifies the hypothesis because otherwise the experiment could just prove that the cheetah is faster over a 2-meter period, but that doesn't really show any endurance on the cheetah's part. So the prediction says that the experiment results are going to show that the cheetah will be faster than the lion over a 60-meter period. Then comes the test portion of the scientific method. This is very important. This is the experiment part of the scientific method. This is when the scientist either proves or disproves his hypothesis. So I'm going to write here that the scientist is going to use a radar gun. So the scientist is going to point the radar gun at a cheetah running over a 60-meter period and write down how fast the cheetah was running, and then point the radar gun at a lion running over a 60-meter period and write down the speed of the lion. Then comes the analysis portion of the scientific method. This is when the scientist looks at the results and decides whether they prove or disprove the hypothesis. So in this case, the analysis is going to prove and confirm the hypothesis. So I'm going to write here that the hypothesis is confirmed because the results of the experiment showed that indeed the cheetah was faster than the lion. So if my example confused you a little bit, don't worry about it. The important thing to remember is the main points of the scientific method: the question, hypothesis, prediction, test, and analysis.